Okay, so welcome to this next video in which we are discussing the adaptive immune response. Okay, so at the moment we're looking at the humoral immune response. Okay, so we've just generated a B cell that has a fantastically good B cell receptor as far as binding to this antigen is concerned. Now, it's going to come out of this affinity maturation process, and it's now going to differentiate. And it can either differentiate into a memory B cell, or it can differentiate into, well actually I haven't shown it here, but it differentiates into a cell type known as a plasminoblast first, okay? And then what will happen is this plasminoblast will divide and divide and divide and divide, and then the offspring, the daughter cells of this plasminoblast, will then differentiate into plasma cells. Okay, so we'll start with what memory B cells are for. Memory B cells will remain in B cell follicles, but as I say, they're not stuck in B cell follicles. They can go into the blood, they can circulate for a little while, they can go into a different lymph node, sit in the B cell follicle, they can also go to the spleen and other lymphoid organs. So B cells are not static, they're not stuck in a single lymph node. Okay, and the point of memory B cells is that if the infection is to return again, then what will happen is potentially this antigen will go up in the blood. So you'll get lots of this antigen in the blood, and this will bind to the B cell receptor on the surface of this memory B cell. So let me show this here. Here's the B cell receptor on the surface of the memory B cell, and the antigen will come along and bind to that, and it will then activate that memory B cell, and this memory B cell will now divide and divide and divide. Well, it will differentiate back into a B cell. It will divide and divide and divide to make many B cells, which will then lead to the production of a huge number of plasma cells. So the secondary immune response proceeds much faster. There was none of this faffing about with the somatic hypermutation and affinity maturation in germinal centers within lymph nodes. There was no need for T helper 2 cells. It just went for it, basically, and produced a huge number of plasma cells on the spot. And this is the real reason, these are the real reason that the secondary immune response is so much faster, because you've got memory B cells sitting there, and all they need to see is this antigen again, and they'll then just go off on one, basically. They'll return back to B cells and produce a huge, great population of plasma cells extremely rapidly. Now, what do these plasma cells do? Well, they produce antibody. Okay, now what does antibody do? Well, it's going to attack the extracellular pathogen. Okay, so there are two options here. Either the antigen that the antibody binds to, and by the way, the antibody is a special soluble form of this B cell receptor. It's the B cell receptor, but without the final bit that attaches it onto the membrane. Okay, so they secrete this soluble form of the B cell receptor known as an antibody, and either the antigen we have put this reaction up against is a secreted antigen, and therefore potentially could be a secreted toxin, or it could be on the surface of the microbe. Now, if it's a secreted toxin, then the binding of this antibody to that secreted toxin can serve to neutralize the toxin. Whereas if it's a surface antigen, then the binding of the antibody to that surface antigen can kill the microbe in two ways. Firstly, the antibody can serve as an opsonin, so it can bind to receptors on the surface of phagocytes such as neutrophils and macrophages, and it basically makes the neutrophil or macrophage more likely to phagocytose this microbe. Alternatively, the antibody can recruit the classical complement cascade proteins and lead to the destruction of this microbe through those complement cascades. Okay, so that is how antibodies are effective against extracellular microbes. So that now covers the humoral immune response, which is totally about the production of these antibodies which are going to attack the microbe. We'll now turn our attention to a cell-mediated response. Okay, so in the cell-mediated response, then the pathogen was intracellular, okay? And the famous example of an intracellular pathogen would be a virus particle inside the cell. Okay, right. So, the thing you have to understand about the cell-mediated or the intracellular pathogens is that they're not always intracellular. In order to spread between cells, they have to temporarily go extracellular. So, basically you're going to initiate all of the stuff you initiated for the humoral immune response, but you're also going to initiate a whole new ballgame. So, basically, when the intracellular pathogen is extracellular, i.e. when it's spreading between cells, then it can be phagocytosed by these antigen-presenting cells again. And when these antigen-presenting cells phagocytose that pathogen when it's extracellular, they will then present the antigens which they get, well, the antigen fragments which they get by breaking down the pathogen. They'll expose them on MHC class 2 again. Okay? They will then go through the same process. They'll migrate to the lymph nodes and they will activate CD4 positive T cells. Okay? And these CD4 positive T cells will then differentiate into T helper 0 cells. And this time, when you get a whole population of T helper 0 cells after this initial T helper 0 cell has proliferated, these T helper 0 cells will start differentiating this time into T helper 1 cells. And again, the process by which you communicate to the T helper 0 cells that the pathogen is intracellular and therefore that they need to form a T helper 1 response rather than a T helper 2 response is not understood, but we know that interleukin 2 plays an important role. Okay, now what do these T helper 1 cells do? Well, basically, they start secreting interferon gamma, or IFN gamma for short. And interferon gamma is basically a horrendous signal to macrophages. It's basically like the, I want you to bring no prisoners. I'm not interested in prisoners, basically. Take no prisoners. It's a horrible signal that the T helper 1 cells send to the macrophages to tell them that they can use brutal tactics, basically. Don't worry about being nice, just go nasty, basically. Fight fire with fire, basically. Okay, so it turns on a huge number of macrophage killing tactics that weren't on before, basically. It makes the macrophages more powerful. It gives them new powers. Okay, so what's the use in this? Well, two very famous examples are when we're dealing with mycobacteria and listeria species of bacteria. These are intracellular pathogens that live within macrophages. So, when the macrophage engulfs mycobacteria or listeria bacterial species, what will happen is they'll go into a phagosome, and then usually what should happen is that lysosomes should come and fuse with this phagosome and release lysozymes, which are very powerful destructive enzymes. They should release them into the phagosome, and these lysozymes should then destroy the microbe within the phagosome. Okay? However, mycobacteria and listeria species, of which the main examples for mycobacteria would be Mycobacterium tuberculosis, the causative agent of TB, and for listeria it would be Listeria monocytogenes, which causes gastroenteritis. Okay? These basically prevent the fusion of these lysosomes with the phagosome. They prevent what's known as phagolysosomal fusion. Okay, and they then live within the phagosome and divide within the phagosome perfectly happily, basically. Okay, now this is not good. Basically, what interferon gamma does is it gives these macrophages more power. It helps these macrophages destroy these intracellular pathogens, basically. Okay, so that's the purpose of the interferon gamma. Now let's look at the other portion of the cell-mediated response. So, this is the part that's totally different from the humoral response. So basically, if your pathogen is intracellular, then the pathogen may well actually infect the macrophage without being phagocytosed. So for instance, if it's a virus, the virus might just infect the professional antigen-presenting cell, this dendritic cell or this macrophage. And then the pathogen will be within the cytoplasm of the cell, but it will not be within a phagosome. Okay, so you'll have all sorts of antigens from this pathogen that are within the cytoplasm of the cell that weren't even there because it had been phagocytosed, but were just there because the pathogen was inside them. Okay, and then what happens is the cell will break down these antigens and express them this time on MHC class 1. So to express things on MHC class 2, you have to have got that antigen via phagocytosis. To express things on MHC class 1, you just have to have this thing within your cytoplasm. And all cells do this. This is nothing special about macrophages or dendritic cells. Every cell that has a nucleus in your body will have MHC class 1 protein complexes on its cell surface membrane. And it will be continuously breaking down proteins within the cytoplasm into fragments and mounting them on MHC class 1. Okay, now, what's special about these professional antigen-presenting cells is that they will also be activated by the pathogen-associated molecular patterns of this intracellular pathogen. So the PAMPs will activate the pattern recognition receptors within these dendritic cells and macrophages, and that will lead to this professional antigen-presenting cell also putting co-stimulatory molecules on its surface, such as CD8 and either B7.1 or B7.2. Then what will happen is the professional antigen-presenting cell will go from its initial site to a lymph node, and it will percolate through the cortex and the paracortex. And this time it's going to be looking for a CD8 positive T cell. Okay, so one of these guys here. Now, again, CD8 positive T cells, they are all different. They sit within the cortex and the paracortex, and you'll have a huge number of them, and they all have different T cell receptors on their surface. So the antigen-presenting cell needs to find a CD8 positive naive T cell which has a T cell receptor that is capable of binding to this specific antigen fragment. Okay? When it finds this perfect CD8 positive T cell, then it presents the antigen fragment on MHC class 1 here to the T cell receptor, and that T cell receptor is complementary to the MHC with this antigen fragment, and then CD8 will also bind to the MHC class 1. This delivers signal 1 to the CD8 positive T cell. Then the co-stimulatory molecules, CD8 and B7.1 or B7.2, they will activate their receptors. So CD8 will activate the CD8 ligand, and B7.1, B7.2 will activate CD28. This will deliver signal 2. Then this cell will undergo the same process that the CD4 positive T cell underwent, i.e. it will release interleukin 2. It will also produce the interleukin 2 receptor alpha, which will combine with the already expressed interleukin 2 receptor beta and gamma proteins to make a full interleukin 2 receptor. The interleukin 2 will then stimulate this interleukin 2 receptor, and that will deliver signal 3 to this still naive CD8 positive T cell. Okay, right. Then what will happen is, where am I? Here. The CD8 positive naive T cell will then differentiate into what's known as a cytotoxic T cell, and it will then divide many times to make a whole population of cytotoxic T cells. Okay, now what do these cytotoxic T cells do? Well, they go into the blood and they're going to cause apoptosis of infected cells, and that's not what I want. Where's the final piece? Here. Okay, so all over your body you will have cells which are infected with this intracellular pathogen. Okay, so let's say it's a virus. You have cells that are infected with the virus, and they therefore have this antigen within them. Now, as I said before, all cells in your body which have a nucleus are capable of antigen presentation. So they continuously chop up proteins that are within their cytoplasm and present them on MHC class I protein complexes on their surface. So basically, cells which have viruses or this intracellular pathogen within them will have MHC class I complexes with this antigen fragment on their surface. The cytotoxic T cells will come along and bind to those MHC1 complexes with the antigen fragment. So the T cell receptor will bind to the antigen fragment and the MHC class I, and then the CD8 will bind to the MHC class I. And then this is all you need. You don't need co-stimulatory molecules, because remember, these normal cells aren't capable of making those co-stimulatory molecules. Okay? All you need is the MHC class I with the antigen fragment. And then what will happen is the cytotoxic T cell will cause this cell to commit suicide, basically, to undergo apoptosis, and it's induced via the Fas ligand, or the extrinsic apoptosis pathway. So this causes this cell to die, basically. So it's going to go around and kill cells which have got intracellular pathogens within them, and this is a protection mechanism. You're going to sacrifice a few cells to save the whole body, basically, to save the other 100 trillion cells. So one cell will die, well, a few cells will die to protect the whole meta-organism. Okay, and those are the two portions of the cell-mediated response. One of them is through the CD4 positive T cells, which go through the T helper 1 pathway, and the other is through the CD8 positive T cells, which is a branch of the adaptive immune system that's only activated in response to intracellular pathogens and not to extracellular pathogens, and it causes the apoptosis of cells which have the pathogen within them. Okay, thank you.